I did my research presentation on the article Vasoactive intestinal polypeptide expressing interneurons in the hippocampus support goal-oriented spatial learning. This article studies the relationship between polypeptide hormone, vasoactive intestinal polypeptide, to goal-oriented spatial learning. More specifically, this study examines the role of vasoactive intestinal polypeptide in the neurocircuitry which allows for spatial learning. The schematic of this circuitry involves two interneurons, allowing for disinhibition. A disinhibitory interneuron is responsible for the activation and inactivation of another interneuron. In context to this study, the disinhibitory interneuron activates or silences another interneuron, which then activates or silences a pyramidal cell within the CA1 layer of the hippocampus. The CA1 area of the hippocampus can be seen on the figure to the right. The pyramidal cells in the hippocampus can also be referred to as place cells. Place cells represent certain locations in space and therefore are responsible for cognitive mapping. The underlying circuitry and processes of how these spatial representations are formed and changed is not completely understood. Additionally, a simple random spatial representations may have different firing patterns than goal-oriented spatial learning. Therefore, I decided to examine the associations between hippocampal circuitry to goal-oriented spatial learning through a molecular lens. Previous findings have results supporting that disinhibitory interneurons are present within the hippocampus and have a role within learning. In addition, former studies have shown that one subpopulation of interneurons express vasoactive intestinal polypeptide, or VIP. These VIP-expressing interneurons are disinhibitory neurons. The basic schematic between these VIP interneurons, other interneurons, and pyramidal cells has been studied. The first part of this specific pathway starts with a VIP interneuron. The second part is characterized by another interneuron. Finally, the schematic ends with a pyramidal cell. Now, if the VIP expressing interneuron, which is the first cell in this schematic, was to be firing, the second interneuron would be inhibited, which would allow for the pyramidal cell to fire. In contrast, if the VIP expressing interneuron, which is the first cell in the schematic, was not firing, then the second interneuron would be firing, which would be inhibiting the pyramidal cell. With the structural knowledge gained by these previous studies, I conducted this study to more closely observe the firing patterns of the interneurons and place cells. As shown in the figure, the second picture depicts place cells which fire at specific locations in space. I wanted to specifically examine the role of VIP interneurons within this schematic. Furthermore, I wanted to investigate the relationship of VIP interneurons to the behavior of goal-oriented learning. This study will allow for the comparison between goal-oriented spatial learning and random foraging. The differences between firing activity of VIP interneurons for goal-oriented spatial learning and random foraging will allow for a greater understanding of the overall role of VIP interneurons. This experiment consisted of 3 experimental groups, all having different effects on the VIP interneurons. VIP interneurons activation was modulated by the use of optogenetics. Mice had VIP interneurons tagged with ARCH-T, tagged with channel rhodopsin, and they have mice not tagged with anything. The mice first went through 9 sessions of running on the track with no manipulation. After 9 sessions, the mice then went through another 9 sessions with the presence of light. Therefore, it activated the ARCH-T and channel rhodopsin tagged VIP interneurons. The ARCH-T tagged interneurons were being inhibited by the ARCH-T, and the channel rhodopsin tagged interneurons were being activated. In addition, this study used mice models. The mice were trained by running on an oval-shaped track and given a water reward at one specific location for each lap that was made. The number of licks the mice made at the reward location were recorded each trial as a measure of their memory of the location. Furthermore, my study utilized in vivo imaging with the use of a 2-photon microscope. Pyramidal cells were labeled with GCAMP, which is a calcium binding site that upon binding to will fluoresce green. VIP interneurons were labeled with TD tomato, which fluoresces red. This can be seen in the figure to the right. The results of my study showed that mice were activated VIP interneurons while learning the goal-oriented location of the water reward had an increase of learned response in comparison to the control group and group with inhibited VIP interneurons. This increase in learned response was noted by the increase of licking when at the reward location. In contrast, the mice who had had their VIP interneurons inhibited by the ARCH-T had a decrease in licking when at the reward location. Furthermore, the place field distributions for the VIP control had an increase in activation near the site of the water reward. This can be seen through the heightened activity near the reward in the last session versus than in the first, as pointed out by the arrow. In contrast, the results of the VIP ARCH-T tagged interneurons who were inhibited shows no significant difference in the place cells firing. Previous findings defined the structural aspects of learning by defining the disinhibition aspect. The study was able to further add to the knowledge of disinhibition and specifically be able to identify the changes of firing patterns with goal-oriented learning versus random foraging. Goal-oriented learning had higher firing rates within place cells during the location of the reward. Overall, this study supports that vasoactive intestinal polypeptide is necessary for goal-oriented spatial learning. The results showed that while ARCH-T was activated by optogenetics, therefore inhibiting vasoactive intestinal polypeptide expressing interneurons, it caused there to be no spatial learning. In addition, when channel rhodopsin was activated by optogenetics, the vasoactive intestinal polypeptide expressing interneurons were activated, which caused there to be an increase in spatial learning. Therefore, a key part to spatial learning is the expression of vasoactive intestinal polypeptide in disinhibitory interneurons. Furthermore, spatial learning with a purpose or goal-oriented learning showed stronger firing patterns in pyramidal cells than foraging did. This shows vasoactive intestinal polypeptide may also play a role in the strength of firing in disinhibitory interneurons, which allows for a stronger versus weaker firing pattern. This can also be depicted in the figure on the screen. The knowledge gained about the role of vasoactive intestinal polypeptide and disinhibitory interneurons can be applied to future studies of other learning and memory research. Since not much is known about the processes of learning mechanisms, these additional findings can be a foundation for other researchers to continue. Since there are many subtypes of interneurons within the hippocampus, learning more about vasoactive intestinal polypeptide expressing interneurons gives more insight to the possible roles of other interneurons present. As shown on the screen, there's lots of different interneurons characterized by different morphologies, connectivities, and markers. The paper was well organized in a way that established validity of all of its methods before showing results for the experimentation. By testing all the methods with known knowledge, the internal validity of the experiment was checked efficiently before using methods on the experimental groups. In addition, the figures in the paper well modeled the findings of the study. However, I felt the middle of the article, in particular, was not organized very strongly. The middle section of the paper is split into many subsections surrounding different ideas, and it came off as choppy. That being said, overall, I believe the paper did an efficient job at setting up its ideas, checking its methods, and being detailed about the experimentation.